Good evening. I am Saurabh Sevla. So today I am going to discuss about the photosynthesis. This is the syllabus of class 10. So in this topic, we are going to cover following subtopics like what is photosynthesis, how does it take place, what are the stomata, what are the function of stomata, and activity necessary for the photosynthesis. Right? So we start. What is photosynthesis? So photosynthesis is an anabolic process. It is an anabolic process in which the fixation of carbon dioxide with water in presence of sunlight inside chlorophyll of leaf to produce carbohydrate and liberate molecular oxygen in the air along with water. So this is the anabolic reaction. It means the simple reaction convert into complex product. So carbohydrate is a complex product and C6H12O6 is a glucose, and such glucose further convert into sucrose, and sucrose further transfer into the starch by the polymerization of the glucose unit together with the chemical bond. So after that we have the structure of the leaf, and leaf is the site of photosynthesis in the green plants. And the photosynthesis take place is in all green plants as well as certain cyanobacteria, right? Not all bacteria, because some bacteria are heterotrophs, but cyanobacteria are showing the autotrophic nutritions. So they are known as blue-green algae, and they are the part of photosynthesis. So in this structure, the leaf is actually dissected into two parts, upper epidermis and lower epidermis. So the upper epidermis bear waxy cuticles. These waxy cuticles actually prevent the water loss. And below this, the mesophyll cells are there, and they bear the large number of chloroplasts. And beside this, air cavities are also there. And on the lower surface, there are stomata present, and stomata play a very important role, like exchange of gases, transpiration, and photosynthesis also. So now the events of photosynthesis, the photosynthesis reaction completes in three major steps. So first, the absorption of lights by the chlorophyll, and in this process, the light is absorbed by the chlorophyll, and chlorophyll bear the thylakoids. These thylakoids are the coin-like structure. They are called grana, and they have the photosystem, means they have the pigments which receive the sunlight, and such sunlights bear light energy, and these light energy converts into chemical energy, and this chemical energy formation is called photophosphorylations. Simultaneously, the splitting of water molecules which take place inside the lumen of the thylakoids, and during splitting of water, the water molecule breakups into hydrogen and oxygen respectively. So that's why oxygen produced by the plants in daytime, in most of plants. After that, at night, the reduction of carbon dioxide. It means the carbon dioxide is added with the hydrogen in the stroma part of the chloroplast to produce carbohydrate or glucose. But there are some exceptions like desert plant. These plants actually take up the carbon dioxide at night and prepare their intermediate products. But they absorb the sunlight in daytime also through the chlorophyll. So now the activity shows that what are the raw material of photosynthesis. So we know there are four raw material of the photosynthesis. One is sunlight, second chloroplast, third carbon dioxide, and fourth water. So these raw material actually represent the necessary parts of the photosynthesis. So we discuss one by one. So first discussion, that is, what is starch? This is the one of the important question. So starch is called the carbohydrate. It is a complex carbohydrate or polysaccharide, and starch is known as the reserve food of plant. So starch actually stored by the when large number of glucose unit linked together by the chemical bond. So this activity actually shows how starch is produced. So in activity A, it is actually showing that the leaf is kept in the boiling water. It tries to be the decolorize or solubilize some parts. After that, it is transferred to the B part, and here the leaf is kept in the test tube, and it is also present in the ethanol. So ethanol is the organic solvent, and it decolorize the leaf. After that, the leaf is transferred to the one of the working area, and here you put one of the iodine, one or two iodine drops, and iodine drops shows the blue color. So presence of blue color means there is a starch present. So it is the general activity which we are going to discuss in the following another activities, right? So first activities, that is sunlight necessary for photosynthesis? So in this, we have taken one leaf structure and it is covered with a black strip. So on strip area, we say we assume that there is a no sunlight fall on such leaf area surface. So after some days, we remove these leaves, and it is kept in the boiling water. After that, transfer to the ethanol or alcohol solution to make that decolorize. So after decolorization, it has shown that some part which are total white and some part which are decolorized, and we put one or two iodine drops at different area. So the surface where strips are covered, so no blue black coloration take place, as well as on the another side the blue black portion found because presence of starch. So it clear that sunlight is necessary for photosynthesis by this activity. Okay. So in the next activity, this activity actually shows, is chlorophyll necessary for the photosynthesis? So in the photosynthesis, we know it is take place in the chlorophyll. So we took one croton leaf or money plant leaf. So it again decolorized by the ethanol, and we put the iodine drops on the leaf surface. So area the on the leaf which is green, so it shows the blue color, otherwise no blue color. So it also show chloroplast is necessary for photosynthesis. So activity is very simple, and this is one of the important activity as per the CBSE pattern. The next activity, that is carbon dioxide is necessary for photosynthesis. So carbon dioxide, it is the major part because it take place in the reduction process. Okay. So for proving the carbon dioxide of the photosynthesis, we took two potted plants, and both are kept in the tight jars, and they are also sealed. So before sealing up, in one of the activity, we kept the potassium hydroxide solution. But in B activity, there is a no potassium hydroxide. So role of potassium hydroxide is very important. It absorbs the carbon dioxide. So if the carbon dioxide cannot be reached to the plants, so we say that there is a reduction of carbon dioxide process will be affected. So no photosynthesis, no glucose formation, or no starch formation will be there. So finally, the iodine test is failed in activity A. But if we discuss in activity B, all raw material are there like sunlight, chlorophyll, carbon dioxide, and water. So no potassium hydroxide solution were there. So it unable to absorb carbon dioxide. Therefore, the carbon dioxide is totally captured by the plant's leaf through the stomata, and it can be fixed along with the hydrogen and to form carbohydrate. So this activity is very important, and it shows that carbon dioxide is necessary for photosynthesis. So now come back to the leaf structure. So on the lower epidermis, there are leaf stomata, and these leaf stomata play a very important role. So First, they are teeny pores. They are teeny pores, and the pores have bean shaped, dumbbell shaped found in monocotyledons plant, but in dicotyledons leaf, the shape of guard cell is beans, and they are made up by guard cells, and they are surrounded by epidermal cells, and they have stomatal pores. So they have three major functions. First, stomatal pores helps in transpiration. It means water comes out from the leaf and it convert into vapors form. This is called transpiration. Second, exchange of gases. During daytime, stomatal pores release oxygen and absorb carbon dioxide. Okay. And third one is the photosynthesis, because guard cell bear few chloroplast. So these are the function of tiny pores. But the next question is that how are stomata open and closed? So it depends upon the water activities as well as the potassium ion. So when waters from guard cell, the waters move from epidermal cells to the guard cells, as well as potassium ion also comes from epidermal cell to the guard cells. So stomata get open due to the endosmosis, and most of the stomata are open in the daytime, but in night there is a no photosynthesis take place. So that's why the potassium ion comes out from the guard cells to the epidermal cells, as well as water do the same activities. So water molecule also comes out from guard cell to epidermal cells due to the hypertonic environment, presence of hydrogen ions inside the guard cell, which make the abscisic acid. So the stomata get closed. So these are the activities, how stomatas are open and closed. So they depend upon the water movements through the osmosis, right? So in last, there are certain practice questions. Question number 1, what do you mean by starch and how it detect in the plants? So you know starch is the complex carbohydrate. They are called the polysaccharide, and they are detected by the iodine solution. On a starch, when we put 1 or 2 drop of iodine solution, so we say it shows the blue black color. Otherwise, uh, if the solution like cellulose, glycogen, if you treat the iodine solution with them, so you may not get any kind of blue black appearance, right? Question number 2, why potassium hydroxide used in experiment for CO2 in the plants? So it is because potassium hydroxide absorb carbon dioxide, and in the following experiment we used to study CO2 is necessary for photosynthesis. That's why we use potassium hydroxide. Question number 3, name the plant's leaf which has less chloroplast. So there are two major examples, croton leaves as well as money plants. Okay. Question number 4, how stomata are open and closed. So stomata are tiny pores. They are found on the lower epidermis. They open when water moves from epidermal cells to the guard cell through endosmosis, and they close when water comes out from guard cell to epidermal cells at night. So these are the functions or activities. Question number 5, draw well-labeled diagram of stomata. So stomata are bean-shaped structure found in the lower epidermis of dicotyledons leaf, and their functions are three. First, photosynthesis. Second, exchange of gases like oxygen and carbon dioxide. And third is transpiration. So thank you so much from my side. Have a nice day.